So with that, I'm prepared to hand over the screen to you, Ramesh.
Okay, yeah, let me see if I can share my screen here.
I'll stop my share. Looks good.
Looks good. Okay. Okay, great. Yeah, just thanks everyone for attending. You know, what I wanted to do today is just kind of give like a little short tutorial about what carbon nanohoops are. And I wanted to show you a little bit about a, you know, not a brand new project, but certainly a newer direction in our lab is trying to develop these into actual things that might be useful as biological imaging tools. Okay, so let me get a pointer here. All right. So the name carbon nanohoops is derived from, I guess, you know, from their relationship to the structure of a carbon nanotube. Okay, so on the left here is a, you know, a picture of a carbon nanotube, you know, which is a hexagonal carbon structure made of entirely sp2 hybridized carbon, or you can think of it as made of all benzene rings, right? Linked in a way that you make a cylinder. So if you take a small slice of a carbon nanotube, let's say just one layer of it, you can see a structure as the one that's drawn here. That's what we kind of refer to as a carbon nanohoop, just to separate it from a nanotube. And so this is an interesting molecule. You know, these molecules were actually proposed. You can, you know, track it down all the way to the 1930s. Certainly in the 80s and 90s, physical organic chemists were interested in making this structure completely aside from carbon nanotubes. I mean, at those times, you know, carbon nanotubes had not even been discovered. So someone envisioned a structure where you have a pi system and it's linked into a ring like this. And they're like, oh, that's a really interesting structure, but nobody had ever been able to make something like this. And the reason is, you know, if you look at this molecule, okay, so the acronym in brackets here is NCPP. So N is the number of phenylene units in a cycloparaphenylenes. Okay, so it's a all phenylene macrocycle that's linked in the para position. Okay, because of this geometry of the structure, each phenyl ring must be non-planar. But in sophomore organic chemistry, right, we all learned that aromatic rings, and especially a benzene ring, has to be flat. But, you know, carbon nanotubes are made with this basic structure, but they're made at, you know, around, you know, elevated temperatures, 800-900 degrees, right? So making a structure like this using organic chemistry seemed daunting. And you can look at these numbers here. So 6 cycloparaphenylenes would be a nanohoop made of just 6 phenylene units. The number here, 96 kcals per mole, is the strain associated with that molecule relative to a linear version of itself. So by bending the pi system from a non-planar thing, you have to account for 100 kcals per mole of extra strain energy. So you know, that's not a trivial task. And so, you know, as you make the larger rings, the strain becomes a little bit less, right? Because it approaches more of a less bent phenylene unit. So this was the challenge. So these kind of molecules kind of remain dormant for a long time. And I want to show you, so, you know, obviously we won't be talking about this if we weren't able to make these. So I don't want to go into a long discussion of how we made these, but I just want to give you an idea of how we put these molecules together, or nanohoops together, okay? So the key is, you know, you can't take a string of phenylene units and wrap it into a ring. It's just going to be too costly energetically. So we came up with a different way of making these molecules. I'll show you just one example to kind of highlight how this is done. So basically here, what we have is we have like a biphenyl unit that we oxidize. Okay, this oxidation actually creates an sp3 center in one of what will become a phenylene unit. So then what we can do is we can add other nucleophiles to build the structure up. So for instance, we can add 2 more phenylene units. Okay, and then you can keep doing this where you can deprotect the silyl group, and then you can oxidize it again. And now you can add another nucleophile. Notice that we're building up a structure where, this unit here is basically a cyclohexadiene it's an oxidized phenyl unit, okay? But the 2 oxygens are sent to each other, which is important in order to eventually, we're going to close this into a macrocycle. So I'll show you this in the next step. So you see here, we just closed the macrocycle using the Suzuki coupling. This is a synthesis that we developed back in 2012. Actually, we can make these in much higher yield now. But the basic point here is the reason that you can make this macrocycle is because the cyclohexadiene is not an all phenylene unit, right? It doesn't have all sp2 hybridized carbons. So it can be bent such that you can wrap it into a ring. Then the trick was, how do you convert that then into a benzene ring? Well, as I mentioned, these cyclohexadienes are essentially oxidized benzene rings. So if you use a reducing agent, like this was a single electron transfer reagent that you can use, this will actually convert the cyclohexadiene to a phenylene unit. So you can make these all phenylene macrocycles, even ones that are highly strained, and they're stable once you make it, they're kinetically stable, and you can isolate these things. And so this is the way, we made many different types of diameter structures of these carbon nanohoops. And the beauty of the synthesis, I think, which is different from, the typical nanomaterial, is that it's a stepwise synthesis. So essentially, if I want to add, let's say I want a ring that's not just a normal phenylene ring, I could cross-couple in something else. Like I could use a pyridine ring, or I could use a different type of aromatic ring. So the nanohoops actually are broad ranging in their type of structures. And I'll show you that in a second here. But this is the basic idea of how we make it. So we can make, hundreds of milligrams of these different types of molecules, over, 6-7 step type syntheses, that type of thing. So here's, I think a range of these structures that just differ in size. Okay, I think, the first thing that becomes really apparent, when we made these, is that they had this weird property that they were redshifting in their emission as they got smaller. Okay, I mean, this is exactly opposite what you would normally think. You usually think that if you add more pi conjugation, you're going to redshift the emission properties. But in this case, if you see, the HOMO-LUMO gap is narrowing as you make smaller structures. We can go into why that is later in the discussion if you want to, but this is completely opposite to nanoparticles or linear conjugated systems. And we observed this, and we knew this afterwards by, also calculations and various other things. But this was kind of surprising and interesting feature that we learned right away. Okay, so what's cool about the optical properties are, somewhat interesting and unique about these is that you see this redshifting emission. You see this redshifting emission, but notice that all of them can be excited at the same wavelength. So this was kind of unusual based on looking at the HOMO-LUMO gaps. You would think that, the absorbance should also redshift as it gets smaller, but it does not. And the reason is because these molecules are centrosymmetric. So actually, the HOMO-LUMO transition, that excitation, is forbidden based on symmetry rules. And so what happens is these, this major absorbance here, that's usually around 340 or, closer to 345 nanometers, is actually a combination of, like say the HOMO to LUMO plus one or things like that. And so those transitions are actually basically identical for the different nanohoop sizes. So you can excite all of them at the same wavelength, and they will emit different colors depending on the size. And so what happens in the emission is, in the excited state, there's a breaking of the symmetry, which allows the emission to be, which gives it, the proper symmetry such that it can actually fluoresce. Okay, and so you see, blue to green, and it'll go to yellow eventually. But as you probably can't tell completely from here, but actually, as you go smaller and smaller, the quantum yield drops. Okay, so this is also kind of an interesting feature. So in the first larger structures, the quantum yields are around 90%. If you go towards the middle, maybe like 8 rings around or 7 rings around, the quantum yields can go to like 10%. Still relatively bright, but there's a trend there. And this is because as the structures get smaller and more rigid, that symmetry breaking event in the excited state doesn't happen. And therefore it's not allowed to fluoresce. And so you don't see any fluorescence. So it's kind of cool that because we can make these from the ground up, we can alter this, we can change this. So for example, here's a all para Nanohoop. This one has 7 rings around. It's essentially non-emissive. If I take one of these phenylene units, and instead of it being paralinked, I make one that's metalinked, which we can use the same synthetic procedures to make. Now, all of a sudden, this is now fluorescent because you broke the centrosymmetry. So, with nanotubes, the optical properties are all controlled by that fine detailed structure of how they're linked, how the carbon atoms are linked, but you don't have that great synthetic control on those things like we have here. And so I think this is just an example of how you can manipulate the optics. So this is in this paper down here below. So there's other things you can do. For example, as I was telling you, you don't have to make these molecules with just phenylene units. For example, here's one with a benzothiazole structure. The reason we made this one is because this is a very electron accepting unit. So it should lower the LUMO and redshift the emission. So we make this molecule using, again, basically the same procedures. And we look at the emission, and you can see it still has the absorbance around the same place, but 10 CPP is basically a blue emitter. And now it's in the orange by just having these 3 atoms appended to it. You can see in comparison with a normal just 10 phenylene structure, you've redshifted the emission by 100 nanometers, but you haven't actually done anything to the absorbance, which is kind of cool that you can still excite it at the same place. We kind of explored this computationally and have a good understanding of why this stuff is the way it is. And we even wrote a paper recently in chemical science where we are just calculating structures that nobody's ever made, but looking for things that might be interesting. For example, this molecule we calculate to have emission in the near IR. So, these can actually be pushed out pretty far into the red. It remains to be seen whether we can make all these types of structures, but I believe it's possible. So this gives you an idea of how you can, yeah, is there a question?
Yeah, sorry, I didn't mean to interrupt, but wanted to find a time here. I want to encourage any questions in the chat window. And if there's anything I can do to, help, encourage that, questions along the way. I was just going to submit a question around the extinction coefficient. So 10 to the 4th, what is that? How broad is that extinction coefficient range across the different sizes or, essentially colors here?
Yeah.
Is there any way to turn it up?
Turn it up. We made polymers of these structures, like where these are actually the monomer of a longer polymer. We have some structures are around 10 to the 6th. I think that's about as high as we've seen. If you get larger, the extinction coefficient goes up some, but there's a limit to it. I'm not sure. We haven't actually played around with that a lot. It's a good question.
Anyways, so I had a question. I wanted to use that to encourage others to post questions in the chat. So.
All right.
Good. So I see Dimitri just asked a question. What's the limit of resolution on the tuning?
The limit of the resolution, meaning on the emission,
Yeah.
presumably. Yeah, I don't know what the limit is. You know, there are some fine tweaks that one could make to, you know, to move things around by 20, 30 nanometers. I'm not sure. I don't have a good feel for that, what the limit is.
Okay, that's why we call it research, right? Good.
Indeed.
All right, sorry to interrupt.
Yeah, no problem. Okay. Let's see. So I have the chat window up now, so hopefully I'll actually see it if somebody asks something. Okay. So anyway, so this is you know, an example of being able to tune the emission, and also, you know, highlights the fact that Nanohoop started out as like a fragment of a carbon nanotube, but I actually think putting in other types of aromatic units and, you know, making new types of structures that way actually is quite interesting for applications. But let me go to the slide here. Okay. Let me do one thing. There we go. Okay, so, you know, you have these interesting optical properties, and you know, presumably maybe this could be used for some type of biological imaging, but these things are all, you know, hydrocarbons, right? How are you going to ever dissolve this in water? And, you know, can you actually use it for that purpose? And so, you know, we had this idea for a long time. It actually took many, many years to finally get to something where, you know, we had made something that was water soluble by using these sulfonate groups. So this is an example of it's 8 cycloparaphenylenes. It has 8 phenylene units. We pretty much made it the same way. You just have a substituent on one of the aromatic rings. It sounds simple, but it was actually took a lot of time to figure this out. But the cool thing is this molecule with just 2 sulfonates is now water soluble. You know, it's not immensely water soluble, but enough to do some imaging experiments. So in this first paper, which we did in collaboration with the Pluth group, you know, we made this molecule. We're able to treat cells with the molecule and see if it goes inside. It does go inside. It's the green fluorescence that you're seeing. It seems to be kind of spread out all through the cytosol. You know, the optical properties are maintained in, you know, biological media, which we were not sure about. We actually also made a version of this that could target a specific cell type to just show that that was possible. And so this was the first example of us kind of like, okay, you can turn these into fluorophores that could operate in a biological system. And so, you know, there's some limitations here. One of them was, you know, that 340 nanometer excitation, you know, is okay for some things, but maybe not the best thing for, obviously, for imaging living cells. So we wanted to address some of these things. We also thought maybe there's simpler ways to target, you know, these things to even intracellular components. So we followed this up recently, which, you know, hopefully this paper will be out soon. Where, and again, this is in collaboration with the Pluth group. And then we also worked with people at OHSU, the non-group, to do some more advanced imaging, actually. So what we're able to show is here we make this is actually a relatively dim nanohoop compared to other nanohoops because it's a smaller size. But we have this meta linkage that makes it, you know, it turns it on at least a little bit. But what we did is we clicked on, so we use a alkyne substituted CPP, and we just click on linkers. And those linkers we can even use to attach targeting groups. For in this example, we added this morpholine targeting group, which has, you know, been used many times to localize things to the lysosome. So we just wanted to see, you know, can we get this into a cell? Will it, you know, localize in the lysosome? Can we see that? And what we wanted to do in this case is use 2-photon excitation. So instead of having to excite it at 350 nanometers, you could excite it at 700 nanometers if it will be something that has a good 2-photon cross section. I mean, everything is not that way, you know, so you have to find this out. So, you know, what we did is we measured the 2-photon cross section, and we, incubated some cells, and we looked at these things. And so, without going to a huge amount of detail, yeah, you can excite these with 2-photon. You can traffic these to certain places in the cell, and you can image them. So that was exciting for us. So let me go to the next thing. Maybe I'll come. Let's see. I see a question here. Permeable to live cells, is it possible to functionalize? Yeah. Okay, so for sensitivity to ions or metabolites, I'm going to talk about that a little bit, about how you can get, maybe sensing different type of things in a second. So maybe we can come back to this later. But we kind of established that the optical properties are tunable. There's probably a whole range of things that could be done. And it looks like you can get them into cells, if you have the right functionality and et cetera, et cetera. And so one of the things we're thinking about is like, that's great. Okay, it's a dye, it can go in, but how do you take advantage of maybe there's some other unique features that you can use. So one of the things that's interesting is these are really strained molecules, right? So that's kind of one of the things that I told you about at the beginning that's unusual about it, and what made it, kind of a dormant class of structures actually for a long time. So that's one thing that I thought that there might be a way of taking advantage of that. The other thing is, most fluorophores are not macrocycles. So, it kind of makes you naturally wonder whether there's some ways of taking advantage of like host guest chemistry, supramolecular chemistry. And I thought that might be a way to provide more function to the fluorophores, like sensing capabilities. And I'll show you that in a second here. Okay, so I think, we're pretty familiar, I think all of us with, or a lot of us with click chemistry, right? Reactions that can, that have high fidelity. I mean, the prototypical click reaction would be an alkyne reacting with an azide with some copper catalyst, right? So you can take two entities and put them together in a really high efficient reaction. Kind of more modern version of that is the strain promoted azide alkyne cycloaddition reaction. So that one has been used, in biology immensely, because basically if you have an alkyne that's it's strained, it's bent, it's so reactive, you don't need the copper catalyst. And that's important because then in a biological system, you could have a click reaction that could occur and you don't need that copper to be in the system. That's actually quite useful. Probably some of the most, I think, well-known examples, and the person that kind of developed this kind of concept is Carolyn Bertozzi in this, bioorthogonal chemistry, where, meaning that you can do chemistry in the context of, say, a living cell, and not disrupt what's naturally going on. And so, a classic example would be where you take a cell and you feed it some sugars that normally are displayed on the cell surface, but instead of the natural sugar, we have an azide attached. These things then get incorporated and displayed on the cell. The processing machinery doesn't realize that it has a functional group on it because the azide is so small. And then you click on these cyclooctines, and therefore I can find out where those sugars are, where do they go. And so this is a good example of it. And these strained alkynes have become so ubiquitous in biology research that, of course, you can look on Thermo Fisher's website and you can buy these type of reagents, like this one, the Dibo reagent. It's not a cheap reagent to get, but in these kind of experiments, you don't really need much. And so this is an example of where strain is used in probe design. Okay, so in 2018, we made a series of nanohoops that have an bent alkyne in the backbone. And, we're wondering whether, because we have such good syntheses of how to control the size, and that these are fluorescent molecules, we thought maybe you can make clickable nanohoops, where you could change the optics and you can maybe modify even the amount of strain and the reaction rates. We made these and we quickly were able to see, yeah, they still have the fluorescent properties. They still have the size dependency. The question was, will they undergo those kind of reactions, or, will it turn into a mess, right, if you expose it to some type of reagent? So the first thing we actually did, so we were at that time, we were not thinking about bioorthogonal reactions too much. We're actually thinking about more electronic materials. And so this reagent TCNE will do a click reaction. This is a 2+2 with the alkyne, then it actually ring opens. Okay, so it is a reaction that has been used before. We didn't discover this reaction or anything, but we were curious whether it could be promoted by strain. And what we saw is that, yeah, if you use a bigger one, it's still strain promoted, so the reaction proceeds pretty quickly. But if you decrease the size, you can get it all to go all the way at 0 degrees. These are high yielding reactions. So, prototypical click type reaction, but this is not the type of reaction that's used in biological systems. We're wondering more recently, can you do this with an azide? And, is it going to be so much slower than cyclooctane that it's not really useful, or maybe it's comparable. There's not many scaffolds that have these like strained alkynes besides the cyclooctane. So we started now looking at the rates of these reactions. Let me move this thing. And so this is the reaction rate with benzyl azide. Okay, so we monitor these reactions, get the reaction rate. If you want to think in comparison to cyclooctane, one of the very early versions of cyclooctane, is around 10 to the -3. There are better versions of that reagent. The Dibo one is the one that was in the catalog that I showed earlier. But we thought, well, can we manipulate these structures and change the rates? So for example, what can you change, like put withdrawing groups on here, right? So we put some electron withdrawing groups and that accelerates the rate. Another trick that we were able to play is like, if you make one ring across from the alkyne meta, it puts a lot of strain on the alkyne. And you can, we have ways to calculate those kind of things to the strain, but that accelerates the rate. So, kind of undergoing, looking at all these things, we realize there's so many possibilities of structures, which rings are meta. What if you go 8+1? So we call it, let me just quickly explain the nomenclature. We say 11+1 if it's 11 phenyl rings plus 1 alkyne. So, we know that 7+1 might even go out 0 degrees. 8+1 should be faster. So it just seemed like a huge amount of things to make. So we actually started collaborating with some computational people on the Lopez group at Northeastern. And, they have calculated structures for us where the rates would be super fast. It's a matter of can you maintain selectivity and can you actually make these kind of things? But I think the range is, I know, convinced me that we should continue looking at this because we can actually get to a wide range of rates, rates that would even beat, the most common reagents used today. So this is like an ongoing thing, but, you might imagine that you could also think about changing the colors of these. So like if you incorporate, that benzothiazole unit, you can make these kind of reagents too, and you can still shift the optics around. So, we're kind of thinking that this could be, usually you would have a cyclooctane and then some fluorophore hanging off of it. Now the fluorophore is the strain component. So I think there's some room to do some interesting things there. And so this is kind of, the early days of this, but we're excited about this. So let me show you one last thing, then I'll get to the questions. Hopefully, I'll be brief in this part.
Yeah, we're gonna have to do a time check, but it was very interesting. So thanks.
Yeah, okay, I'll keep this part quick. So let me just show you the one last thing. So supramolecular chemistry. So again, most fluorophores are not macrocycles, right? So, one way to make, I think, to be able to add some function into these structures is to make mechanically interlocked molecules. Those are types of structures that are, basically the simplest type of molecular machines, but you can add some function to these. And what we did is we came up with a way where you can actually click two components together in the cavity of the hoop, and then it links those two in the middle, and the molecule cannot escape the macrocycle because the two ends are big enough to hold it in, okay? And the hoop is so small, you can see in the crystal structure that that's a very compact structure. So the reason we made this one is that dinitrobenzene is a quencher, so it quenches the fluorescence. So just as a proof of principle, we thought, let's add fluoride, okay? If you add fluoride to this non-emissive molecule, it should break the oxygen silicon bond, and then the quencher floats away, and then it should turn on, right? So that works beautifully. You can do that. And so that was kind of a proof of principle, but we knew nobody really cares about sensing fluoride. So, we wanted to go to something else, right? So just most recently, we showed that, and this again is working with the Pluth group. Mike is, one of the experts in the world of trying to understand what the role of H2S is in the body as a signaling molecule. So we thought maybe we could put one of his probes and lock it into the middle of this molecule, and that maybe you will end up with some selectivity on that reaction. Usually those reactions, may react with H2S, but it could react with other thiols as well. They're also good nucleophiles. So in this case, we thought that maybe the rigid hoop surrounding the probe will give you selectivity for small analytes. So, this is kind of a little bit of a guessing game, but this worked better than we would have ever thought it would work. So for H2S or, HS minus with this counter ion, the fluorescence turns on immediately. But if you take a T-butyl thiolate or even benzyl thiolate, where the neighboring atom is just CH2, it's not huge, neither one of those turn on. And looking back in the literature, it makes sense because rotaxanes like this, where you have a hoop surrounding a thread, has been used to protect structures on, that are threaded through. So it kind of makes sense that you would get selectivity. But this is cool. And so, I think, this is pretty much the last science slide. So the idea is that you could develop like a general sensor design. The triggers can be different, and the payload could be different. But, the basic chemistry is that the hoop is a fluorophore, so you can kind of read it out when the event happens. So those are the early days of these type of things. And so, we're interested in always collaborating with others. Let me quickly highlight the people that did all the work. So starting from the oldest people, or people that have been gone the longest. So Brittany White was a graduate student here. She made the first water-soluble hoop. Terry Lavelle was a graduate student here. So Brittany's at Cornell now as a postdoc. Terry was a graduate student here as well, now a postdoc at McGill. She did all the 2-photon stuff, the meta hoops, the redshifting hoops. Toby is a professor now in Germany at Heidelberg. He developed the strain cyclooctines that now Julia Fair is kind of taking over. And then Jeff Van Rayden developed how to make those mechanically interlocked molecules. And then Claire is the one that did all the work with the H2S to show the selectivity. And so that's it. We're happy to have these collaborators and obviously money. And thank you for your time.
Very good. Very good. Thank you. I did have a couple of questions. So I think Dan Beecham asked if the probes were stable to fixation or cross-linking in cells and imaging.
We haven't tried that. I mean, I would assume that, I guess it depends on what type of cross-linking one is thinking about, but
probably formaldehyde.
Yeah, I mean, it should be. I mean, I don't think, these things are not very reactive.
Right.
Unless
you design them, right, to be, right.
Yeah, so they're very stable.
So someone asked a question about how, if the rings always stay in plane with one another, or they start to spin or go perpendicular as the hoop gets larger. Like where's the sweet spot where things start to spin?
So it's difficult to tell. I think the smaller ones, I think it's very strained for for a phenylene to rotate in the middle because you have to contort the molecule. So those, I think the smaller ones probably do not flip through like that, but they can still wobble. back and forth. And the larger ones, I believe they should be spinning. But we haven't done a lot of NMR work or anything to really tell what the barriers are. This is partially hand waving my.
And then I have one last question. I know that Yu-Zhong is ready to present, but one question about a multispectral reagent. I'm not sure how that would work. Dimitri asked this, wondering if you could get 2 emissions from a single ring.
It is possible. We've seen some structures like that. For example, we weren't intending to do that, but we've appended some, fluorophore or whatnot, and sometimes you will see emission from 2 things. We put 2 hoops together, and you can sometimes get emission from each one.
Okay.
Yeah, so it is possible. We even have interlocked 2 hoops, and you can see emission from both.
Oh, that's really neat. All right. Well, I really appreciate it. Great presentation, really interesting.
Thank you.
In the interest of time, I'm going to hand over to Yu-Zhong. So Yu-Zhong, I don't know if you can grab the screen and share yours, or if Ramesh needs to.
Yes, I can do it. By the way, Tony, so it looks like I have 7 minutes. Sure. So
we reserved for 15 minutes afterwards too. So 15
minutes. So
yeah,
I will go quickly, maybe show a few slides.
Yeah,
I know everyone has a tight schedule. So first of all, let me share my screen.
Yep.
Can you see them? No?
Not yet.
Not yet. Can you see them now?
Yep. There we go. Yep.
Good.
Good
By the
sign.
way, I really enjoy the talk of Professor Jasti's talk. I'm glad that I have another, colleague is working on non-biological nanoparticles. So my talk today is also on non-biological particles, but it's slightly bigger. It's in micrometer size and use it as a calibration standard to improve the data of flow cytometry analysis. So let me go quickly. For Thermo Fisher Scientific, we have a very comprehensive technology portfolio for researchers doing the flow cytometry. I think my colleague have already discussed many of them in the previous talks, but today I will focus on some not mainstream product, but we call them microspheres. So today I just try to give you a very brief review about these microspheres and why do we need this. These microspheres mainly a synthetic polymer particles. Mostly used are polystyrene particles, with or without cross-linker. And based on the applications, these can be plain white particles or loaded with single or multiple fluorescent dyes. So why do we need this particle, and how can they help us to obtain high quality data? So I think that to better understand, for me to better explain it, we need to look the components of flow cytometer. So the flow cytometer, I think you may all well know. So I just very briefly mentioned they have 3 major components: the fluidics, the optics, and the electronics. They work together to provide a complete system for cell analysis. So in this case, the director in today's talk, I want to emphasize that the detector is one of the critical components of the flow cytometer. The most commonly used, as we all know, is the photo multiplier tubes, called PMTs, that reside in each channel of the instrument. You can see that there are all the PMTs. So some of the instrument, of course, they use APD. We can discuss that later. A PMT detector captures the photons that are emitted by the excited fluorophores, either on the surface of cells or on particles. Of course, they also has a forward side scatter signals, and they convert them into a photocurrent, which is then processed to the electronic system. So actually the initial signal, the light hitting the detector, is amplified within the PMT to improve the sensitivity and the resolution of the measurement. And the second is the signal amplified is controlled by the PMT voltage. And in most fluorocytometer, this voltage setting is a user adjustable setting. So this is an advantage we can see, because when you use adjustable PMT, if the signal is low from negative, you can increase the PMT to get a better separation. If the signal is too bright, by lowering the PMT voltage, the signals will remain in the linear range of the detection. That sounds like a big advantage. But in the same side of thing, it also become an inherent weakness or shortcomings of flow cytometry measurement from the quantitation standpoint. So let's see an example why this become a problem. So here is a fluorescent intensity measurement. I just did an example. I pull a 6 micron C7-like particles. It's stained with our special dye inside of the particle. And if I just do the regular flow cytometry measurement, when I set up the PMT is 200 milliwatts, what I got reading is 16,866, about 17,000. So is this value also will be changed in case I increase the PMT voltage to 240 milliwatts. It increase about 5 times, or if I decrease the voltage to 160 milliwatts, it's also decreased. So this complexity of the instrument design and the signal amplification system has resulted in many types of inter and intra-laboratory variations. So that's all depends on the PMT setting, the measurement of the so-called mean fluorescent intensity of a given sample in day 1 could be different from day 2, or also could be different from different labs. So this example, I think, could tell us again, this advanced fluorescent technique like a flow cytometer demands quality at every level. Not only just specimen preparation, fluorophore selection, panel building, but on the final step of our very long experimental protocol, the data collection is critically dependent on the instrument. So one way to resolve the problem and to improve the accuracy and the reproducibility of the measurement is to use controls or calibration standards. One of the most used reference are the fluorescent microspheres. So again, I'll just quickly go through. I think that a lot of you may already used the fluorescent microsphere before. Why we use this non-biological particle as a reference? Why we just use cells? Because cells has a lot of bio variations in terms of size, in terms of the surface marker density, in terms of the labeling variations. But the microparticle, if we use polystyrene or any polymer particles of well-manufactured particle, we can have a particular given size. For example, just like the cell size, what we can make in here in Eugene, we can make the microparticle up to 20 micron. We also can make a small nanoparticles like a 20 up to 200, 500. And they are highly homogeneous. We can put either single or multiple fluorescent colors or color combinations with controlled emission intensity. And especially they have as another advantage is a long-term physical and photochemical stabilities. So they are all way better than use biological samples. There are many types of fluorescent particles used in calibration, not only flow cytometry, but also imaging. And we have a lot of product for imaging system calibration as well. But today, I mainly just focus on the fluorescent intensity reference based on the example I gave to you before. So you can really measure the fluorescent intensity independent from the voltage setting. And but what type of fluorescence dye to use and how to assign the value of the intensity of the reference particles. So that's a big question. Instead of just develop some type of reference by our own, we choose to work with the National Institute of Standard and Technologies because they are the leader in setting the reference material. This work start is a long history and complicated, so I just mentioned this very briefly. You have to first start to select different fluorophores. Before actually 2002, there is no standards in fluorescence measurement. What is a unit? What's the intensity? The people are just talking about the bright, dim. That's not a quantitative measurement. So first of all, we work with NIST from 1999. It started to select. First is a fluorescent, FITC, the widely used. Then we get more dyes to cover the spectrum. Recently, you can see on the table until this year, we introduced more dyes to add a so-called NIST reference material. And use the NIST reference material. Let me talk this quickly. We just load this dye into the microspheres, and the scientists at NIST help us to value, to assign the so-called equivalent of reference fluorophore value to the nanoparticle, to the microparticles. It's like you develop a ruler which inches or which meters. It's the same idea. But since for the flow cytometer, they have so many different excitation sources and the emission channels. So currently, we have this NIST traceable equivalent of reference fluorophore reference material for 26 channels, have 3 intensity peaks. And it enable the fluorescent intensity measurement for instrument accuracy and sensitivity. So here is our product. We just released a few months ago. So it's easy to use, and we measure it. You can see this is just a screenshot of our atom flow cytometer workspace. You can see they emit in these, atom has 14 different channels. You can see all of them, and it's well separated. So here we also gave a customer a table of the fluorescent intensity, and I will quickly show how to use it. So it's very easy to use. If you have a flow cytometer, if you have a sample, you just need to, there's 2 ways to do the so-called ERF value assign calculation of your own sample. One is the simple way called the nearest neighborhood reference method. You just put 1 drop of your sample and also put 1 drop of the ERF beads in the same tubes. Then you run them together in the chosen filter, which your cell sample will emit. In this case, we have the FITC filter. Then you calculate you can see these cells in the red peak. The intensity is pretty close to the medium intensity beads in the reference standard. So the reference medium intensity reference beads, the MFI value, the intensity value is 8,725, and the cell sample is 13,000. So it's 13,000 divided 8,700. So you just time this one with the known value, ERF value, 80,000 of the medium beads. You can easily calculate that is 121,000 of my cells. That's one way. Another way is you generate a standard curve. I'm not going through this. I think it's easy. You just, first you run the beads, you get the value, then you run your beads, then you use the standard beads value to generate a curve. Then you calculate the ERF value of the test sample by cross calibrating its MFI value against calibration graph. So that's another way to do it. But so now we go to a technical summary. I mainly want to say these ERF beads with needs assigned ERF value will enable the so-called quantitative standardization of a fluorescent intensity scale of the performance of flow cytometers. ERF value also will help you to assign your samples value, but you have to make sure you use the value in the table in the particular fluorescent channel or say filter site. This ERF particle, the third thing is you need to pay attention, is only fluorescent intensity unit reference. The biological characteristics of cells, such as the density of surface marker or level of biomarker expression level, cannot be measured by this product alone. You need to have a biological controls. So you use both the biological control and the Accu check ERF beads, then you can assign or examine the level of biomarker expression in your cell sample. And also to answer to support the what do you call that, the global fight with SARS pandemic, we also collaborating with NIST to develop a nanoparticle scaled ERF beads. That's not only for the intensity, but also for concentration. So where now will be developed. And this one will support virology and extracellular vesicle research. And I think, I know at least in some other side of the Thermo Fisher Scientific, a lot of people doing the extracellular vesicle research. If they want to have the sample or you want to develop some collaboration, we will looking forward of that. And thank you again for your attention. So today we focus on the fluorescent intensity reference. I hope in the future in your research, you can use more of the calibration standard will help you to improve your data accuracy and reproducibility. Thank you.
Great,
that's it.
Thank you.
Sure.
Really nice summary of a lot of work, Yu-Zhong, and a great collaboration across between Thermo and Thermo Fisher Scientific and NIST. Love to see that connection. Do we have any questions for Yu-Zhong or for Ramesh? I know we set aside some extra time, but I don't know that the meeting invitation includes that. I know that mine actually ends at 2.
Yeah, mine also ends too.
Yeah, so, but I do appreciate, thanks for trying to abbreviate your presentation, Yu-Zhong. In the interest of time, if I don't have any other questions, I do want to thank everybody for attending. And whether you had a question today or question from that you asked in the chat, or you have a question you'd like to follow up on, my understanding is that this has been recorded, and so we can refer anybody that would like to see the recording to the link. And I certainly encourage everyone to reach out to Yu-Zhong or Ramesh or to other attendees with any follow-up conversations. The intent of this meeting is to first of all to educate one another on some of the areas of interest, but also to introduce and promote collaborations. And I think there's a lot of opportunity from both of these presentations, and I certainly want to encourage it. So, all right, with that, I think we can end the call today. And I wish everybody a happy Wednesday.
Thank you, John. Thank you, Tony.
Thanks.
Thank you, everyone.
Thank you.
All right.
Thank you.
All right. Have a great day. Thanks. Bye.